third video on oxidative phosphorylation. In our previous two videos, we have discussed about the basic concepts of electron transport chain, like what are the complexes and how the electrons get transported from this complex 1 and complex 2 to ultimately complex 4 and finally to oxygen, and how this proton gradient is generated, how the ATP synthesis is associated with the ATP synthesis. So we have discussed this ATP synthesis procedure, the structural integrity of ATP synthase, and how the rotation of gamma subunit causes the structural configuration changes of beta subunit, and how the ATP synthesis ultimately works in our second video. So the links will be in the description box. Also, you can see on your screens, I think, during this time, or maybe at the end of this video. So anyways, please, if you're new in this channel, please go and check those videos. I think that they will really help. And today we'll be discussing about this chemiosmotic theory, which is very, very important theory. But before that, let me just okay. So today we'll be discussing about this chemiosmotic theory. So this particular theory was first given by Peter Michel. He was the scientist, he was the brain behind this concept, and he also won the Nobel Prize in Chemistry in 1978 for discovering this chemiosmotic theory. And he proposed that a proton motive force is actually responsible for driving the ATP synthesis through ATP synthesis. If you can remember, in my previous video, I have mentioned one thing, not in here, but in here. Yes, I have mentioned one thing, that the F1 ATP synthase subunit can perform the ligase function, that is the ligation of ATP and PI, without the proton flow into the matrix. However, the release of ATP, requires the flow of proton through the membrane. This is very important. So, based on this fact, this proton motive force, the concept of proton motive force is working or was discovered. So, Peter Michel first stated that proton motive force is responsible for driving the ATP synthesis. And the proton gradient as well as the membrane potential are the proton motive force that is used to drive the ATP synthesis. Again, the same thing that I have already mentioned in my previous two videos, that there are basically two things. One is the chemical potential, that is pH is more. pH is less in this intermembrane space, and pH is more in this matrix. Okay, so since H plus concentration is high in this intermembrane space, pH is less. So acidic pH in this intermembrane space, and since H plus concentration is low in the matrix, so pH is high, and so basic pH or basic condition, basic environment in this matrix. This is associated with the chemical potential, that is pH, delta pH. That means pH gradient means inside alkaline. Obviously, outside acidic, inside is alkaline or basic. And another thing is electrical potential. Electrical potential means since H plus ion is accumulated in this intermembrane space, so this part will be positive and this part will be relatively negative to this intermembrane space. So positive in the intermembrane space and negative in the mitochondrial matrix. So this is the electrical potential. So inside is negative and outside is positive. So you can see over here, pH inside is delta pH, that is pH gradient, inside is alkaline, and delta psi, that means electrical potential, inside is negative. These two things actually help in the ATP synthesis driven by proton motive force. And so due to this electrochemical gradient, that means gradient in the electrical gradient and the chemical gradient as pH, delta pH, this electrochemical gradient is actually associated with the ATP synthesis through this ATP synthase. Okay, because if there is no gradient or no gradation produced due to the accumulation of H plus, there will be no inward movement of this proton or no inward movement of this H plus through this ATP synthase, and so there will be no ATP synthesis. So that is the chemiosmotic theory, and which was proposed by Peter Michel, and he won Nobel Prize in Chemistry in 1978. So this is very, very, very important theory, chemiosmotic theory. Now let's deep dive into the uncoupling agents. Very, very interesting agents are these uncoupling agents. So uncoupling agents are those which can, uncouple the electron transport chain and the ATP synthesis or ATP synthesis. So as we can see in here, that these two are kind of related. So this is like ATP synthesis, electron transport happens in this part, electron transport happens in this part where complex 1, 2, 3, 4 are associated, and ATP synthesis happens in this part where ATP synthesis is associated. But there are some uncoupling agents which can just break the link between these two processes, that is electron transport system and ATP synthesis. So they can break the link between the electron transport system and the ATP synthesis. So what happens when uncoupler is present is the electron transport system will go on. Electron will get transferred from NADH or FADH2 to oxygen, and oxygen will get reduced to H2O, but it will stop the ATP synthesis. So ATP synthesis will not be there. ATP will not be produced, but electron transport chain will remain intact. Okay, so this is the structure of uncoupling agent, and this is basically dinitrophenol. Okay, so you can see this is the structure of a phenol, and this benzene ring. This is the structure of a phenol with this OH group, and dinitrophenol because there is a two NO2 group. So this is one NO2, this is another NO2. So this is dinitrophenol, which is a very, very, very common example of uncoupling agent or uncoupler. So in presence of uncoupler, what happens is in presence of the uncoupler, electron transport continues, but no ATP synthesis occurs. So why this is so? Because they they see, as I have already mentioned, that for this electron transport system, this proton accumulation is not required. This proton accumulation or the proton pump is required for the ATP synthesis. Okay, for ATP synthesis, rather to release the ATP. So what happens in this case is the uncoupling agents or the uncouplers will inhibit the accumulation of this proton in the intermembrane space. Okay, that is the thing. And they instead they will they will not produce ATP. And this when this uncouplers are present, there will be no production of ATP, but there will be production of heat. So heat will be generated. Okay, so uncoupler electron transport system continues, but no ATP synthesis occurs. The second point is dinitrophenol is a weak acid that is hydrophobic enough to soluble in the inner membrane, and as a result, it is protonated in the intermembrane space and deprotonated on the matrix side of the membrane. Because no ATP is made, energy from the food is not available for the fat synthesis, and dinitrophenol was previously used as a diet drug until their side effects, including liver toxicity, was proved. Okay, so what happens? How this dinitrophenol actually works is this is very interesting because the thing this is the inner mitochondrial membrane, and dinitrophenol can inhibit the formation of this proton gradient across this inner mitochondrial membrane. Okay, because it can go easily to the intermembrane space, and from this intermembrane space it can go easily to the matrix because this is hydrophobic and very weak acid. So it can cross the plasma membrane easily. So what happens is whenever it is present in the mitochondrial matrix, it will be deprotonated. And whenever it is present in the intermembrane space, it will get protonated. Okay, so it is protonated in the intermembrane space and reprotonated on the matrix. So what happens is, let's say there is a dinitrophenol and in the intermembrane space, and it gets protonated. That means it will use these H plus ions to get protonated. So this H plus gradient will be less, and ultimately there will be no H plus gradient. If we have huge amount of uncouplers, all of these uncouplers will take up these protons that are being transported from complex 1, 3, and 4. They will take up all these protons and they will be protonated. So there will be no proton accumulation in the intermembrane space, and then they will come to the mitochondrial matrix, and then they will be deprotonated. So all the H plus ions will get released in the mitochondrial matrix. So there is basically no proton gradient produced if uncouplers like dinitrophenol are present. Okay, and because no ATP is made, energy from food is not available for the fat synthesis, obviously, and so this dinitrophenol was used as a diet drug until it was discovered that this dinitrophenol is very toxic for our liver, and then it was banned. Now the thing is, main question is, okay, now we know that this dinitrophenol like agents or uncouplers are not that good for us because they they inhibit the ATP synthesis. But is there any advantage where dinitrophenols and or like uncouplers can have some important role? Yes, the answer is a simple yes. Why? Because these uncoupling agents can generate energetically wasteful byproduct, that is heat, that I have already mentioned. And this uncoupling agent, the uncoupling protein, one specific uncoupling protein is thermogenin, which is mainly present in in babies. Also, they are present in very high amount in high altitude natives. Okay, so in those people who are living in a very high altitude, their body temperature should be higher than us, so that they can maintain their normal functionality in that high altitude with very low environmental temperature. So thermogenin, what it does is, well, it inhibits the ATP synthesis to some extent, but instead they produce heat, and that heat kind of help those natives to be to be alive in that high temperature, in that low temperature and high altitude. Okay, like brown fat tissues. These brown fat tissues are generally present in the babies, in baby shoulder, or beside behind the neck, that is present the brown fat tissues, and this brown fat tissues also contain thermogenin protein, which is naturally an uncoupled step. So ATP synthesis is less in babies due to these brown fat tissues, but heat is more. And these brown fat tissues generally they get disappeared as we grow, as the baby grow. So that is the thing. I hope that makes sense. So this is the usefulness of uncoupling agents. Now let's deep dive to inhibitors of electron transport chain. So these are some inhibitors that are very common, very important, and we should remember the names. Like complex 1 can be inhibited by metformin, fenformin, rotenone, and IM156. This rotenone is very important, rotenone and metformin, because metformin is sometimes used as drug which can reduce our cholesterol level, which can reduce our weight. So metformin is one thing, but obviously since it's a chemical drug, there will be some side effects, obviously. So don't take this metformin without prescription. Rotenone is another very commonly asked inhibitor. It is mainly asked in different type of exams. So rotenones can inhibit the complex 1. Okay. For complex 2, we have malonate, 3 NPA, lonidamine, all these things, they can inhibit the succinate dehydrogenase, that is the complex 2. Antimycin A, it can directly inhibit the complex 3. Cyanide, then we have carbon monoxide, all these can inhibit this complex 4. And oligomycin, diboxin, this can inhibit complex 5. Okay. And Gossypol is another drug or another chemical that can inhibit ALDH, that is alcohol dehydrogenase, from which NADH can be produced. So these are some very important, very hugely asked inhibitors that can inhibit different components of electron transport system as well as ATP synthesis. So that is the thing. Now let's see some graphs which are like quite interesting to analyze. Okay, so this is the oxygen consumed, and this is the ATP synthesized, and this is the time. Okay, so this red color is for ATP synthesized or ATP, the function of ATP synthesis. This black color is oxygen consumed. Oxygen consumed means this is the electron transport chain. Okay, so you can see that this is obviously time. So you can see that first, when we are adding ADP and PI, there is neither increase in the electron transport chain or neither increase in the ATP synthesis. That's because ADP and PI cannot directly increase the electron transport system or the oxygen consumption. That's because for oxygen consumption or electron transport system, we need what? Electron carriers, right? Like NADH, FADH2, etc. So adding this ADP and PI will do no good to this oxygen consumed graph or electron transport chain. Now, one major question is then why ATP is not synthesized whenever we are adding ADP and PI? Because I have already mentioned that for ATP and PI synthesis, for the ligation of ADP and PI and synthesis of ATP, what we require is the proton motive force or the gradient, right? You remember this chemiosmotic theory. So this chemiosmotic theory suggests that until and unless there is a electrochemical gradient produced between the intermembrane space and the matrix across this inner mitochondrial membrane, there will be no inward movement of the proton. And for this ATP to be released from ATP synthesis, we require this H plus inward movement of H plus or inward movement of protons, right? So that's why even if we are adding ATP and PI, but there is no electrochemical gradient produced across the inner mitochondrial membrane, so there is no ATP synthesis. Hope this is clear. Now let's see. If we add succinate, so we know succinate is associated with what? Complex 2, right? From succinate to fumarate appears, and then in FADH2 gives 2 electron to complex 2. Now the electron transport system should start working. Okay, and the 2 electrons from complex 2 will be transferred to complex 3 and complex 4. So total 6 protons will be accumulated in the intermembrane space, and now there will be an electrochemical gradient, and then this H plus will come within the matrix to this ATP synthesis, and ATP will be produced. So as soon as we add succinate, there is a bit increase in both oxygen consumption and ATP synthesis, right? And then we are adding cyanide. That means cyanide is what? Cyanide is an inhibitor of complex 4. So if complex 4 is inhibited, that means there will be no further electron transfer, no further ATP synthesis. Because obviously, if complex 4 gets failed, there is only 4H plus in the intermembrane space, and that will not do any good because this 4H plus, it will the ATP will take up, some ATP will be produced for this amount of H plus gradient, but after that the ATP synthesis will be inhibited. So this ATP synthesis has been inhibited. So that is the thing. Remember, this is oxygen consumed means the black line, and black line also signifies the electron transport chain, and this pink or red line, this signifies the ATP synthesis. And in the next, we have, first we are adding this succinate. So whenever succinate is added, some ATP synthesis is okay, but some electron transport electrons are getting transferred. So electron transport chain gets a hike. Then we are adding ADP and PI. The electron transport system is again going on, and also there is some ATP synthesis. Then we are adding oligomycin or venturicin. So this will inhibit ATP synthesis. So ATP synthesis is kind of now there is no ATP synthesis going on, but there is still some amount of electron transport is going on. Then we are adding DNP, that is dinitrophenol. This is the uncoupler. Then we can see that there is no effect of this addition of DNP to the electron transport system, but the ATP synthesis is totally stabilized. There is no ups and downs in this ATP synthesis. So there is totally uncoupled. These two reactions are totally uncoupled. So again, this axis is oxygen consumed. This is the time, and this is the ATP synthesis. So hope you can understand that if we add different kind of substances in different part of electron transport system and ATP synthesis, how the oxygen consumption ATP synthesis will act. So I hope that makes sense. And this is it for oxidative phosphorylation. Okay, here is another thing. Oh, there is another thing which you should remember, that this is the absorption spectra of cytochrome C. You can see that this is the oxidized cytochrome C. This blue color, this is the oxidized cytochrome C, and this red color, this is the reduced cytochrome C. And there are three parts of this wavelength. For reduced cytochrome C, this is alpha, this is beta, and this is gamma. But for oxidized cytochrome C, there is only one peak, that is towards in the gamma region, but that peak is not up to the mark till in respect to reduced cytochrome C. So that is not that much important, but these two graphs are really very important from exam point of view. Okay. So that is one thing. This is it for oxidative phosphorylation. If you find this content helpful, please make sure to like, share, comment, and subscribe. Follow for more. See you in the next video. Bye.